Okay, good morning, everyone. So my name is Karin Sundström, and I'm a cervical cancer epidemiologist, and I'm active at the Karolinska Institute in Stockholm, Sweden, and I'm also active at the Center for Cervical Cancer Prevention at the Karolinska University Laboratory. And I'm going to speak to you today about human papillomavirus, or HPV, infection, and the burden of HPV-associated diseases. And I'd hope with this lecture to give you a bit of an introduction also to the themes that will be discussed more in depth during later parts of this course. So the structure for this lecture is that basically first I will be providing some information about HPV and then describe the burden of disease caused by HPV, both in terms of cancer and some benign diseases. And we will pay special focus to cervical cancer since it is such a major issue of HPV-based concern. Finally, I'll be talking a little bit about prevention of HPV through either screening against this virus or vaccinating against it. So you here see a schematic picture of the structure of the human papillomavirus. First of all, please note that papillomaviruses are found both in animals and humans, and therefore we always denote it by the species, in this case, human. Papilloma means approximately something like a nipple-like protuberance, combined with the aspect of a benign tumor. And the HPVs are capable of infecting skin or mucosal epithelia in the human host. Please note that HPVs have a slow evolution. They do not change host species or recombine. The genome is rather small, as we will see, and is enveloped by a capsid protein. The DNA is double-stranded, and this means it's replicated with high fidelity by the host cell machinery. There are more than 200 HPV types isolated so far, and this is done by different research groups around the world, and then they are sent and tested also by the International HPV Reference Center, which assigns a new number when appropriate. I will very briefly describe the primary products of the HPV genes, and please note that these will be covered more in depth later on during the course. What we can mention already now is that they are divided into genes leading to early proteins and genes encoding so-called late proteins. On the early proteins, we have E1 and E2, responsible for the viral DNA replication and amplification. The genes E4 and E5 and their products also deal with DNA amplification. Please note the specific importance of E6 and E7. These produce proteins that are very potent in interacting with cell cycle control and the human P53 and RB families. And these are the basics of the oncogenic potential of HPVs. The late proteins are mainly responsible for forming the viral capsid. They are of particular importance, not because they constitute the cancer-inducing response, but because they are prime targets for vaccination, as we will see later on during the course. And most of the things I talk about today, you can find much more in depth in the IARC monograph 100B, which I'm referencing at the end of this lecture. So this is just a schematic picture showing you the sequence of the HPV genome, which is circular and around just under 8,000 base pairs long. For the careful of you, you might note that the E3 gene is missing, and this is because in the human papillomaviruses, this is lost, but it is preserved in some animal hosts. But for human purposes, we can disregard it. So I will now move on to describing some characteristics of HPV infection. And first of all, I'd like to say that there are always a lot of questions about how HPV is transmitted. And we need to clarify that mucosal HPV types are mainly spread through sexual contact, even though it is possible to have other transmission routes as well. For example, during childbirth from mother to infant. But they are mainly spread through sexual contact, and this could be, for example, intercourse, oral sex, basically all types of skin to skin, or above all, skin to mucosal contact. Both women and men can contract it, and might not always know, or might even rarely know, because most HPV infections are totally asymptomatic. Cohort studies estimate that a majority of us have actually probably had one of these infections at some point in our lives. Some of us may have had recurrent infections or of different types. However, most infections are cleared within anything from a couple of months to a maximum of 2 years. The prevalence peaks at around 25 years, or the age of prime sexual activity. In some regions, please note, though, that there is also a second peak at around 45 years in women. There are several explanations possible for this phenomenon. Some that have been proposed are, for example, later in life reactivation of a virus, or perhaps changes in sexual habits, a second peak in sexual activity in conjunction with divorce trends, for example. And there's lots of literature on that subject if you're interested in engaging further. I'd like to mention the important established risk factors for infection with HPV. Although, as I said, a majority of us will likely run into it, not everyone will. And the risk factors are above all markers for high sexual activity, for example, early age at sexual debut, a large number of partners. It can also be connected to socioeconomic status, whether you use a condom or not. Condom use can reduce and does reduce HPV incidence and transmission, but not entirely. Multiparity, as having had more than several children, is also associated with both infection and also cervical cancer. Oral contraceptives, smoking, and other lifestyle factors have also been established as risk factors. And finally, I want to mention comorbidities such as immune suppression and also potentially host genetics. For a nice overview, you can refer to, for example, the review article by Toda et al. So, of major focus here, of course, will be the range of HPV-associated diseases. Now, the mucosal HPV types are traditionally divided into low oncogenic risk, called low risk, and high oncogenic risk, or high risk virus, depending on their degree of association with human malignancy. Also, note that there are currently 13 mucosal HPV types classified as high risk or oncogenic, notably HPV type 16, HPV type 18, and HPV type 45. But there are 10 others, as we will see. Now, also note that several HPV types, whereas low risk classified, can still cause papillomas, or, well, actually they are called condylomas in the genital region, that is genital warts. But they do not cause human cancers. And these are notably HPV 6 and HPV 11, which we will return to. Now, the high-risk types that I mentioned are established to cause several human cancers, and they are implicated in several others. But please note that we will here focus on the cancer forms that are major and where there is established evidence to the causality. We may see more cancers added to this list eventually, but right now we will focus on these. And I will be going through the list here: cervical cancer, anal cancers, other genital cancers, and also say something about some head and neck HPV-related cancers. I will, however, not cover skin cancer here, but I note at this point that it has been HPV-related to some extent. I will also go into two disease forms caused by low-risk HPV, and that is external genital warts, or condyloma acuminata, and JRRP, Juvenile Respiratory Recurrent Papillomatosis, which, as we will see, is a disease that affects children, and whereas benign, has a lot of morbidity. Presented in a different way, you here see a schematic pyramid of the burden of HPV-associated disease worldwide. And we can see that at the bottom of the pyramid, we have the most prevalent HPV consequence, which is genital warts, where we see certainly millions of cases worldwide per year. Cervical cancer comes in at a second, with around 500,000 cases per year. There would have been even more if not screening programs in many countries have helped to reduce the incidence. And I've then numbered the other HPV-associated diseases higher up in the pyramid according to decreasing incidence. And in a total, HPV is estimated to cause around 5% of all human cancers worldwide. This is of particular note since, as I will be alluding to later on in this lecture, of course, this carries important implications for the significance of vaccination in potentially reducing this burden. So, a few brief words on external genital warts, since, again, the focus here will be on cervical cancer. Condyloma, or HPV, is a benign disease caused by low-risk HPV. It's not dangerous, and it is self-limiting. That means to say it will heal on its own eventually in almost all cases. However, as you may know, it is associated with physical discomfort and potentially sexual stigma. It is very common, though, among both women and men, and the incidence has been estimated as high as 1 per 1,000 in the peak age group of 20 to 24 years of age. Treatments currently include pharmacological options and/or ablative surgery in bad cases, but typically, again, the disease is self-limiting. So, moving on to cervical cancer, our main focus for today, it is the fourth most common cancer among women. The incidence peaks at around 35 to 45 years old age. Please note that this is earlier than several other HPV-associated cancers. And it is the most well understood and described cancer form in terms of HPV natural history. This is an image of the female internal organs, where we see that the cervix is the lower third of the uterus, protruding out into the vagina, and that has the endocervical canal, which links the vagina to the inside of the womb. On the outside of the cervix, we have the so-called ectocervix. This is clad with squamous cell epithelium, and where susceptible to HPV, may give rise to squamous cell carcinoma, which is the main histological type of cervical cancer. We also have the endocervix, which is a columnar or glandular cell lining, which may give rise to HPV-associated adenocarcinoma, which, although relatively rarer than squamous cell carcinoma, is still a major component of cervical cancer. Please note that there is a hotspot in this area for HPV transformation called the transformation zone, which is an area where different kinds of epithelia meet. And the cellular activity and transformative potential of this zone means that it is a prime spot for HPV to unfortunately develop into cancer. So I'd like to describe and walk you through the different steps that are currently understood in the development of HPV infection to cervical cancer. We can see here on the left that we have sexual activity occurring between a woman and her partner or partners that can lead to either transient HPV infection or persistent HPV infection. This may be influenced, for example, by some of the cofactors or risk factors we have mentioned, including smoking habits, which may support persistence of infection, host immunology, or genetics. Other cofactors such as, oral contraceptive use or the presence of other STIs or perhaps HIV status may also influence the likelihood of persistence of HPV infection. Viral factors may also be of note. For example, the copy number or the viral load you are infected with. in terms of supporting persistence or clearance. In its turn, persistent HPV infections and some of the cofactors we've already mentioned may support the transformation of a normal cervical epithelium to the classical stages of cervical intraepithelial neoplasia, CIN, through CIN1, 2, 3, and to invasive cervical cancer if the woman does not manage to clear the lesions in between. Please note that there is a regressive capacity of CIN. This capacity can be certainly measured at different points and estimated at different degrees. I am here giving some figures from the IARC monograph. This process or progression from normal epithelium, influenced and transformed by HPV, through the classical stages of CIN to invasive cancer typically takes 1 or 2 decades, although examples of quicker progression have been noted. Lastly, I just want to give you this schematic picture of what, if you look on the lower panel, the healthy woman's cervix may look like viewed from below during a gynecological examination performed with so-called colposcopy, where you visualize the cervix with a special colposcopy procedure. And we go from, on your utmost left, healthy mucosa showing no signs of lesions, and then an increasing manifestation of cancer tissue and perhaps in the end even a bleeding and lacerated cervix. However, please note that we have the endocervix higher up in the canal where a tumor might grow without being immediately visible upon colposcopy or from just inspecting the ectocervix. So moving on to today's third form of HPV-related disease, I will now say some words about oropharyngeal cancer. As we already stated, HPV can be transmitted through oral sex and so may be found in several locations in the oral cavity and further back and below in the oropharynx. Note, though, and may thus potentially induce carcinogenesis in this mucosa. Note, though, that there are many different kinds of tumors in this region, and classification is key. Overall, a 25 to 35% proportion has been estimated as caused by HPV, but it will vary very much depending on which locale you consider and which certainty of classification you have in differentiating different cancer forms from each other. Now, cancers in the base of the tongue and tonsils have much stronger evidence of HPV causation, and they're up to 90% have been implicated as HPV related. Again, this is sensitive depending on which population you are studying. Please note that unlike cervical cancers, where many of the high-risk types are implicated and causative, mainly in the oropharynx, we're talking about HPV type 16. Since it is one of the types covered by all vaccines, this may carry important promise for cancer reduction through vaccination. But note, though, that for oropharyngeal cancers, there appears to be 2 different forms, at least, with separate etiologies and characteristics. So whereas one track appears to be very much HPV related, and where the patients are younger and have better prognosis, there's also a second track, which is HPV negative, which appears to be more lifestyle related in terms of alcohol and tobacco use, and where the prognosis is poorer. Now, moving back to female cancers only, in contrast to oropharyngeal cancers, which can of course also happen to men, we are now, if we're considering the slightly rarer HPV-related disease forms, returning to the female genital organs. We will now go lower down into the vagina and also extend out to the vulva and the external genitalia. and see that there are cancer forms here as well that appear to be HPV, or that are proven to be HPV related, but are less understood than cervical cancer. Although it is noted that both vulvar and vaginal cancer has similar pre-stages: vulvar intraepithelial neoplasia, VIN, and vaginal intraepithelial neoplasia, VANE, respectively. These diseases mostly occur in older women, even indeed above 80 years of age. Vulvar cancer is the more common of the two, and around 40% are HPV related, mostly squamous cell carcinoma. Vaginal cancer is very rare, but has been shown to be mostly HPV related. However, it should be acknowledged that it can be difficult to disentangle vaginal cancer occurring at the top of the vagina from a satellite of a cervical cancer, for example, or a vaginal cancer in a woman who has been hysterectomized for cervical cancer previously. So I just want to mention that caveat. But again, it is very rare. Anal cancer is a disease caused by HPV, partially, which is coming increasingly into focus. That is because it's been realized that anal HPV infection is actually very common. It's been estimated at 10% in heterosexual men, more than 50% in HIV negative men who have sex with men. And in women, it may be as common as cervical HPV because of infection spreading from the vagina, for example. And that thus be positive in women of around 10-20%, or even higher, depending on which population you study. The natural history of anal lesions is less clearly understood than the cervical, but again, here we have pre-stages: anal intraepithelial neoplasia, or AIN. And such lesions can affect men, women, and above all, men who have sex with men and or have receptive anal sex. And the incidence is also higher in HIV positive people because of the immunosuppression that may lead to more severe consequences of a prevalence of HPV. It's being judged that around 90% is HPV related, again, mainly HPV 16. And whereas the survival, positively enough, for this cancer form is relatively good, unfortunately, the life quality is greatly affected by the treatment options available. And whereas there are screening methods proposed based on anal swabs, for example, for HPV, which appear to work, it should be noted that there are no clearly, established guidelines for how to best treat the lesions once detected. For example, an anal intraepithelial neoplasia. Because treatment with surgery or laser in the anal canal is invasive, it may lead to a lot of morbidity, and we don't want to overtreat in that area. Surgical treatment of a cervical lesion in women is slightly less invasive because the cervix heals very well. But the invasive procedure of a sphincter muscle or the nearby canal can be complicated. So it's an important thing to keep in mind when considering screening for these infections and precancerous lesions. Of course, if a cancer is manifest, it becomes a different discussion. So I want to also say some words about juvenile recurrent respiratory papillomatosis. A complicated name, a very rare disease, but worthy of special mentioning because of the high morbidity it carries. JRRP, or this papillomatosis, is a wart-like lesion formation occurring in the airways of infants or children, hence the term juvenile. There's also an adult form, but that is not a focus here, as the JRRP form is much more HPV related. It is very rare, with less than 1 in 100,000 affected, but it's very resource demanding due to the high morbidity of the airways. The warts may require repeated ablative treatment and invasive procedures, and the disease is considered chronic and relapsing. It's been attributed and proven caused by low-risk HPV 6 and 11, transmitted vertically from mother to child during birth. And the median age of debut is only 36 months. Thus, it is considered an HPV-related disease with intermediate incubation time. And whereas this is a rare article, most of you may never have heard of it. There's a very interesting new paper in Journal of Infectious Diseases showing that in Australia, after the introduction of a female-only HPV vaccination program, they've actually now seen a reduction, apparently, in this disease among children. So if you're interested, I would very much recommend the references listed here for further reading. So finally, for the disease forms, I will say something about penile cancer. And by now, I think you will recognize the characteristics of this disease. As we have seen in the other locales around the genitalia, penile HPV infection is common among those sexually active. But the consequence penile cancer is a very rare disease. So there are many steps in between. The prevalence of human papillomavirus on the penis varies greatly between sites, which study participants you have, and which sampling methods you use. So it's difficult to give a good figure because studies show everything from a few percentage units to more than 90% positivity. However, we do know that penile cancer is mostly squamous cell carcinoma and appears to have precursor lesions, PIN, penile intraepithelial neoplasia. So it would appear that this disease form shares some HPV-related characteristics. And indeed, the proportion caused by HPV is currently estimated at around just below 1/2. That means there are a lot of HPV-negative cases, and they may be associated with chronic inflammation and smoking. I will now move on from the disease burden and the 7 forms of disease we've characterized to prevention. What can we do about this? In the classical disease prevention terminology, we consider primary prevention as preventing the cause of the disease, and secondary prevention as early detection, and certainly limitation of disease severity. In primary prevention, we have prophylactic vaccines targeting HPV, and in secondary prevention, we have screening for, above all, precursors of cervical cancer. There are, as I've already alluded to in anal cancer, also screening proposed for other HPV-related disease forms. There's also been proposals for oral cavity screening for HPV, but this is very much novel. It is not established yet. Therefore, we will focus here on cervical screening. And since cervical screening has been the mainstay of HPV-based prevention before even the virus significance was known, I will begin by describing that, and then I will move on to vaccination. So we here see that the previously well-established technology for screening of the cervix has been cytology. To the left, you see an image of a cervical cytological smear stained with the so-called Papa Nicolau stain, hence the vernacular term pap smear, upon microscopical examination. And microscopical examination of aberrant cells turns out to have a good sensitivity for pre-stages for cervical cancer, amenable to treatment before it becomes an invasive cancer. It can also detect invasive cancers early on, which tends to lead to downstaging of cancers that are identified in a screening program. However, you can see that the sensitivity is not perfect. It's at 74% approximately, with variation depending on the lab's competence. It may also be higher. But it's universally established that repeated screening with cytology improves sensitivity, which is why most cytological screening programs typically recall women who are normal at a 3 to 5 year interval. To your right, I will now describe a little bit about the new recommended screening method for cervical cancer, which is HPV DNA testing of a cervical sample. So it is the same procedure for the woman involved, but a different test. Instead of staining for abnormal cells and then microscopically examining them, we perform an HPV DNA test and see if there is high-risk HPV DNA present in the woman's cervix or not. Sensitivity here turns out to be very high, around 95%. However, high-risk HPV is very prevalent in young women, and that may lead to considering different ages in which to apply which technologies. For example, here in Sweden, we recently decided to use cytology in women below 30 and HPV testing above 30 years of age in order not to send too many women to further unnecessary workup. Even in women above 30, for example, here in Sweden or in other contexts, high-risk HPV may still be quite common, which means that we typically apply a triage test before referral. This is usually cytology, so that only women who are double positive go on to further colposcopy and possible biopsy. Some algorithms also suggest and are evidence-based that you can immediately refer women positive certain HPV types, 16, 18, and 45, directly to colposcopy. A full review is beyond the scope of this lecture, but suffice to say that there are several evidence-based and positive new recommendations available, depending on context and resources available. I would also like to say that in low resource settings, there are other technologies available, such as visual inspection with acetic acid. To give you an inkling of the effectiveness of cervical screening, I'm here showing the age-standardized incidence rate of cervical cancer across the Nordic countries over the last 50 years. And we see a radical decrease after an increasing trend in the respective countries. We see a radical decrease in incidence after the introduction of cervical screening. So in Denmark, this was around 1970, in Norway around 1975, and slightly earlier in Finland and Sweden, respectively. So I will now move on to primary prevention, that is HPV vaccines, because they are the most recent development. And this is a very strong and positive addition to the prevention battery we'll already have available through screening, such that we can hopefully attack cervical cancer and other HPV-related diseases from two ends. Vaccines were developed in the last decades, and they are based on the major HPV capsid protein L1. This means that the L1 product has an intrinsic ability to self-assemble into so-called virus-like particles, or VLPs. And these VLPs are empty virus shells without any infectious or oncogenic capacity, but with a very strong ability to induce high levels of neutralizing antibodies. That is a strong immune response. So that when the next time the body encounters a real HPV infection, the immune response is ready to kick in and prevent it from taking hold. HPV VLPs based on L1 are mainly HPV type specific, but please note that there may be some degree of cross-protective effect against other types that are included in direct valency. Currently, we have 3 vaccines approved. The first is quadrivalent, HPV 6, 11, 16, and 18, and the indications are condyloma, cervical cancer, vulvar, and vaginal cancer related to vaccine types. We also have a bivalent vaccine containing HPV 16 and 18 virus-like particles, where the indications are cervical cancer and precancer associated with vaccine types, but again with some cross-protective properties. Finally, the newest vaccine approved is non-valent, so considers an increased spectrum of high-risk types with the same indications as those above, but also anal cancer added. And very briefly on effectiveness of HPV vaccination, we have already concluded they are highly immunogenic. It is important to point out that they do not affect clearance of already existing HPV infections, as far as we know, or regression of existing precancerous lesions. Randomized trials have shown that HPV vaccines protect HPV-naive women from HPV infection with vaccine types and or some cross-protective abilities, and also precancerous lesions caused by vaccine types. And in HPV-naive women, the protection is excellent. In women exposed to HPV previously, there's also good protection, but there one needs to consider, depending on population, age group, etc., some different estimates of where the protection lies. But for HPV-naive women, it is very, very good indeed. There have also been a number of population-based so-called real-life studies that have yielded extensive results on effectiveness and safety, extending the findings from randomized clinical trials. And I would here refer you to the lecture held by Professor Joachim Dillner towards the end of the course. So, in conclusion, why does HPV matter? Well, it is considered actually the world's most common STI, or at least one of the world's most common STIs. And a majority of us will have had it sometime in our lifetime. Almost all of us will clear the infection, but some, both women and men, will get a severe disease form such as cancer. And we already stated that actually 5% of all cancers are attributable to HPV infection. However, with the tools we already have available for primary and secondary prevention, we have huge preventive potential already going on and also able to improve. So with successful such programs, we can reduce this burden substantially. And therefore, we in our group, as many others, considers that translational research on access and effectiveness of these methods is a high priority. We're therefore very glad you take this course, and we hope you will be able to share the knowledge and use it in a good way. And we hope you enjoy this course. I leave you with two references that are of particular use for this lecture in terms of their wide consideration of different aspects of HPV infection and disease. And with that, I'd like to thank you for listening.